my name is Supriya Pathak, and today my topic for protein expression presentation is melittin. The content that we're going to deal with is the background, the problem statement, cloning strategy, protein expression and its verification, the activity assay, and the future prospects. So, coming to what is melittin? Melittin is the principal toxic component in the venom of the European honeybee Apis mellifera, and it is cationic hemolytic peptide, and it consists of 26 amino acids with the sequence this. It has several important biological effects and has a relatively low toxicity. Melittin has no disulfide bridge in which the end part terminal part of the molecule is predominantly hydrophobic, while the C terminal is hydrophilic and is strongly basic. As you can see, the problem statement today that we're going to deal with is the antimicrobial resistance. It happens when germs like bacteria and fungi develop the ability to defeat the drugs designed to kill them. Resistant infections can be difficult and sometimes impossible to treat. With the emergence of antibiotic resistance, researchers have recently focused on antimicrobial peptides as potential drug candidates. So the cloning strategy we're going to use is the P pastoris was used as an expression system. We can for cost reduction. E. coli can also be used, but I will not use it as it has several disadvantages, including lipopolysaccharide contamination as well as product toxicity for the recombinant E. coli. The PPIC9 vector was used to transfer the melittin promoter to P pastoris. This is the vector that target vector which was used. It has three important components of alpha secretion signal, the His4 gene, and the alcohol oxidase 1 promoter. The presence of them enabled me to use a minimal medium for expression and to extract the secreted form of melittin from yeast cells. As P. pastoris naturally lacks significant secreted protein, the total protein amount of the supernatant is associated with the recombinant peptide. When a culture medium lacking the protein and peptide is used, His4 enables the P. pastoris to grow in a peptide-free culture medium. As you can see, this was the fragment which I was used from Apis mellifera, and this is the sequence. For the optimization of the fragment, that is the gene of interest, NamGene software was used. The optimized sequence entered the vector using the NOT1 and the ZO1 restriction enzymes. Some other sequences were also added. The ZO1 and NOT1 restriction sites were part of the alpha factor sequence, and which was removed by ZO1 and two stop codons. As you can see, this is the sequence. The annealing temperature and the melting temperatures are mentioned. This is the forward sequence and this is the reverse sequence. This was ligated in the melittin sequence and was prepared using the SnapGene software. This, as you can see, is the map for doing the same. This is the promoter gene. This is the inserting the fragment in the PPIC9 vector. So the PGL digestion product was electrophoresis and melittin was extracted from the gel, agarose gel 2%. To ligate the gene cassette into the PPIC9 vector, a mixture was prepared containing 90 nanograms of digested PPIC9, 30 grams of fragmented extracted from the gel, the agarose gel, and 1 unit of T4DNA ligase enzyme and 1 microliter of 10x ligation buffer. The ligation mixture was then was incubated into for 16 hours at 14 degrees Celsius. The PCR product was then ligated into the vector. To confirm the ligation accuracy, a PCR reaction was performed with AX primers and finally sequencing was done. So the sequencing involved the basic three process, the denaturing, annealing and the extension at different temperatures. For the verification, as I told you, agarose gel electrophoresis was used to see if the PCR product has the correct size. For the transformation of the cells, it is very important as this is P. pastoris. We use the preparation of P IC9 melittin vector, and the vector linearization was done by using the following enzymes like SAC1. It facilitates the integration of the recombinant vector into the P. pastoris. Therefore, SAC1 with 1 microliter enzyme and 1 micro per 1 microgram DNA ratio was used for 1 hour at 37 degrees Celsius. The vector was then purified using the silica bead DNA gel extraction kit. The concentration and purity of obtained DNA were measured by the thermoscientific nanodot 1000 spectrophotometer. So this is the transformation process. Gene pulse apparatus was used for the electroporation. The P. pastoris was taken from the minus 70 degree Celsius stock and was used to prepare the competent yeast cells. It was inoculated to 20 ml of YPD medium and incubated at 30 degrees Celsius and 200 rpm. Shaking for 72 hours was done. The competent cells were then mixed with the linearized PPIC melittin vector, which I showed you in the previous slide, and it was moved to 0.2 centimeter cuvette for electroporation, where the gene pulse apparatus was used, and it was incubated at 4 degrees Celsius for 1. The yeast cells were dissociated from the medium, and the pellet was collected again by washing with cold distilled water and sorbitol 1 molar and centrifuged at 4000 rpm for 5 minutes. Finally, the pellet was dissolved in sorbitol 1 molar at 300 micro ml concentration. For the confirmation, we suspended in the distilled water. For protein overexpression, the cells can be used. The cells transformed by the PPIC9 and the melittin vector could grow on a histidine-free medium as they contain the His4 gene. So the 100 ml suspension was distributed on the minimal dextrose medium with the following chemicals, and it was incubated for 3 to 5 days and at 30 degrees Celsius. And for the removal of the extracellular debris and the material, it was washed with distilled water for the distilled water by 3 times, and the centrifugation was done at 8000 rpm for 5 minutes. The final suspension has the OD 2 to 3, and 1 microliter of that was used in the template colony by PCR by AOX primers. Then the screening was done for selection of highest melittin production. The concentration of methanol, pH, and timer effects were evaluated in Staphylococcus aureus slides, as you can see here. To investigate the methanol, you can see a different concentration of methanols were used, and the inhibition was seen at 48 hours of expression. To evaluate the pH effect, different pHs were tested. Some were tested with buffer, and others were tested without the buffers. The time effect was finally compared at 24, 48, and 72 hours under optimal values of methanol concentration and pH. For protein purification, after this mass production was done, and while maintaining the optimal methanol concentration, pH, and time, expression was carried out in 1 liter of BMM medium. Centrifugation at 1000 rpm for 15 minutes was then performed to separate the supernatant, which is containing the melittin, from the yeast cells. Then Vivaspin 20 was used to separate the proteins larger than 5 kilodaltons. As the melittin is less than 5 kilodaltons, it can be easily separated from in the solution. And to see if to determine the solution concentration, Bradford assay was done, uh, and the solution was lyophilized for storage. For the activity assay, uh, to evaluate the antimicrobial activity of the recombinant melittin, values of minimum inhibitory concentration and also the minimum bactericidal concentration were investigated on 5 gram positive and 12 gram negative bacteria. Both microdilution method using the lyophilized powder of melittin was performed for this purpose. After the, as you can see, after the purification, this production of melittin is cost effective, and it paves the way for further studies and also therapeutic usages. So the growing problem of drug resistance poses a major public health challenge, and so there is accumulated evidence which shows that protozoan parasites such as Plasmodium and Toxoplasma are susceptible to melittin at micromolar concentration. So what it does is it obliterates these pathogens by several mechanisms of actions like disruption of cell membrane, destabilization of the calcium, homeostasis, reduction in mitochondrial membrane potential, and so melatonin like this way can help in the antimicrobial resistance problem. It has different other uses as well, which is if it is melatonin is conjugated with nanoparticles, it gives our it promises a great in different medical appliances, not applications. Not only the approach improve the target, not only this approach improve the target specific delivery of melatonin with less cytotoxicity, but also enhances the proteolytic stability of nanoparticle and several other anti-cancer as well as anti-diabetic properties have been studied of melittin as well. If you have any questions, feel free to email me.